Welcome folks. In this video we are going to look at biology paper 2 for the year 2001, and we have question 1 from section A. Figure 1 shows the parts of a wind pollinated flower. So if you look at pollination, pollination has to do with the transfer of pollen grains from the anthers to the stigma, and after pollination we have the process of fertilization. So as long as there is fertilization, then we are looking at sexual reproduction. So a flower is just an organ that was created for or to carry the process of reproduction in flowering plants. Now, before we start answering, I just want to make a simple clarity. So as you are revising or as you are going through this video, please, the importance or the reason behind why we create these contents is just for you to get the concept. The whole thing is about getting the concept. So you may not find the questions to be exactly the same, but if you are able to get the concept, then you can use that concept, you expand it, then you'll be able to answer any questions that you may have in future. Now we have this structure A. Then if you look at this structure, is for you to master the structure, and this is known as the anther. Now the function of the anther produces the pollen grains. Then the pollen grains are male sex organs of a flower, sex cells. Then for structure B, we can say it's a structure that is holding the anther in right position, especially for pollination. So this is a filament. Then we have structure C. This one receives the pollen grains during the process of pollination. So we have the stigma. Now we also have been given a concept from the first statement. The flower that we are dealing with is a wind pollinated flower. So as we are about to move into answering question B, we need to have that in mind, because there are features or characteristics of a flower that carries the wind pollination. So we have question B. Which two structural features in the diagram confirm its adaptation for wind pollination? So you need to look at the structure. So what can we see? If you look at the position of the stigma, so the stigma is hanging outside the flower. So that is the first point that we are going to give here. So we'll say, if you want, we can say filary. So we'll say filary style or stigma hanging outside the flower. Then for Roman number 2, we can now look at the anthers. So we can just say the anthers are large and loosely attracted to filament, so that any slight air movement may cause the pollen grains to fall on the stigma. So we'll say anthers are large and loosely. So loosely attached. So the reason why they are loosely attached is for easy release of the pollen grains during the pollination. Now we move on to question C. Under question C, explain how the two features you mentioned in B assist the mechanism of wind pollination. So the first one, filary style or stigma hanging outside the flower. So this one we'll say stigma hanging outside the flower creates a large surface area for pollen grains. So as the pollen grains are moving from the anther to the stigma, they need to have a large surface area where they are falling. Then in terms of the anther being large and loosely, that helps in terms of any slight movement of the wind will now make the pollen grains to fall on the stigma. So loosely anthers makes it easy for pollen grains to fall on the stigma. So remember, the stigma is just the female part. Now question D. So question D, which feature prominent in figure 1 is not necessary in an insect pollinated flower? So say feathery. So say feathery stigma hanging outside. So hanging outside the flower. So this one is not necessary, important. Okay, so we move on to question 2. Figure 2 represents the nitrogen cycle. So nitrogen cycle tells us the importance of why the cycle is. Why do we need cycle? So we need cycle to avoid the depletion of certain elements. Now we have certain processes here. Now let's have the first question. Question A, Roman number 1. What is the percentage of nitrogen in the air? So we'll say about 78%. Then what of question Roman number 2? Name the two substances A and B. So if you look at substance A, then we have some arrows. We have death and decay. So what happens to a plant if it dies and it undergoes decomposition? Same applies to the animals, right? So here this A can be, depends, I've seen different information on this one based on the book that you are using. In some books you may find this one as organic matter. Then you may also consider this one as ammonia. Right, then for B, if this one is considered organic matter, then our structure B, this should be nitrites, because the gas, before changing into nitrates, it has to undergo through the nitrates, then from the nitrites into the nitrates. Then the nitrates, these are just the forms of which nitrogen can be absorbed by plants. So nitrogen is never used in form of a gas. So that's a concept that you need to have here. Same applies to carbon. Carbon must be used in form of carbon dioxide. Now we have Roman number 3. Name the three processes represented by the letters P, Q, and R. So P is changing, let's say, ammonia into nitrites, or organic matter into nitrites. So this P is nitrification. Then what of Q? So Q is the conversion of nitrates back into nitrogen. So this is denitrification. So whenever a salt contains nitrogen, changes back into its gaseous form, then the process is denitrification. So what of process R? So let's look at the process R. So this one is the process by which now nitrogen in gaseous form is converted into nitrates. Then the nitrates are absorbed by the plants. So this process is nitrogen fixation. Right, so move on to question Roman number 4. Which process in living animals is represented by the letter S? So where is the S? So S, we are told this one, S is producing this compound here. So this can be excretion. So we just say excretion. So during excretion, there are waste products that are produced which contains an element of nitrogen. Then we move on to B1. Suggest why it is essential to include a leguminous crop in crop rotation. So crop rotation is just an activity that involves changing of crops every year. Like for example, this year if you have an area, if this year you plant maize, then next year you need to change. You maybe plant cassava, then after cassava you consider maybe a leguminous crop. Now these leguminous crops have nitrogen fixing bacteria known as rhizobium bacteria in their root nodules. Then the function of these, they convert nitrogen gas into nitrates, makes the soil fertile. So that's the importance of the soil. Say leguminous crop, or say crops, contains nitrogen fixing bacteria. Okay, so which converts nitrogen gas into nitrates and makes the soil to be more fertile. So it's all about the retention and retaining of soil fertility. Then we move on to Roman number 3. So Roman number 3, what are the 3 advantages of using organic manure as compared to the use of chemical fertilizers? So chemical fertilizers, these are fertilizers that are just man-made. Now organic manure improves the structure of the soil and increases its ability to hold water and nutrients. So it increases its the retention of water and nutrients. So say organic manure improves the soil structure, which retain water and nutrients. So the retention in terms of the soil, is the ability of the soil to keep, for example, water and other nutrients. Then apart from being that, then we can say organic manure releases nutrients slowly into the soil. So for this one, if we are to compare with the chemical fertilizer, so the chemical fertilizer, whenever they are applied, they release the nutrients at once, and because of that, then the chemicals that we need to be taken by plants will be used at once. Then apart from this organic manure, contains a lot of nutrients. Okay, so this is what we can say for this question. Right, we move on to question 3a. 1. What is meant by the term homeostasis? So if you look at homeostasis, it's just the maintenance of a constant or stable internal environment of an organism. So there are changes that occurs without our consent. Changes like the vasoconstriction, vasodilation. Those changes happens so that the body temperature is maintained. Why do we need a favorable or a temperature in a given range? So that the enzymes can work effectively. We know the relationship between temperature and the enzymes. We know that the high temperature may cause or make the enzymes to be denatured, and without the enzymes, life won't be possible. Then the reverse now can make the enzymes to be inactive. So the importance of homeostasis is just there to make sure that the internal condition are favorable for metabolic reaction to take place. So the first question is just what is meant by the term homeostasis. So say this is the maintenance. So the maintenance of constant or stable internal environment of an organism. Now for questions like this one, where maybe you are supposed to define a term like this one. So if maybe my definition is different from the one that you know, my advice is that you can just stick to that definition. So if maybe you spot a difference, I'm not saying your answer is incorrect, no. The definitions can be defined in so many ways, so stick to what you know. So the key point is, as long as we've mentioned the key points like maintenance of constant or stable internal environment, then we are good to go. Then for Roman number 2, name 2 functions of the liver which contribute to homeostasis. So it's all about regulation. So to simplify our answers, we'll just say regulation of blood sugar. So if you look at the regulation of blood sugar, we are looking at the conversion of excess glucose into storage glycogen in the presence of the insulin when there is too much of glucose in blood. Then we know that the reverse is true, and the hormone that reverses this is the hormone called glucagon. So this one changes the glycogen back into glucose when there is less. So in case you didn't know, from now on, whenever we look at regulation of blood sugars, it is also part of homeostasis. So to expand this answer, you can just say the liver converts excess glucose to an insoluble carbohydrate called glycogen. And we know that the glycogen is stored in the liver and muscles. And this one will be used in the time where energy is required. Then for the second one, we'll look at the deamination process. Now for deamination, we are looking at excess amino acids are not stored in the body. So what happens to excess amino acids? They have to be converted into glycogen and urea. So deamination is the changing. So the changing of excess amino acids into glycogen and urea. So this is also part of homeostasis. Okay, so that is our answer there. Then we move on to question B. The graph in figure 3 shows the effects of exercising and immersion in cold water on human body temperature. So for this graph, I recommend that you maybe look for a paper that will help you to see the clear picture. So we have some numbers. There is this 0, we have 5, 10, 15, 20, like that. Now let's look at the first question. So the first question is, what was the body temperature after 10 minutes exercise and 10 minutes immersion in cold water? So after 10 minutes of exercise, want to look at the body temperature. So after 10 minutes, let's go where there is. So body temperature, in this axis we have body temperature. So where is the 10 minutes? So 10 minutes is at this point. So if we move here at this point, then so this is about 37.5 degrees Celsius. So 37.5 degrees Celsius. Then after 10 minutes of cold bath, then the temperature reduces to 36.5 degrees Celsius. Then Roman number 2, how long did the temperature take to return to normal after exercise and after the cold bath? So for the actual graph paper, actual graph paper, we have been given the exercise. So this is the point of the exercise. Then this is the point of immersion in cold water. So according to my graph here, it is just taking 30 minutes. So 30 minutes is my answer here. Then after the cold bath, about 15 minutes. So like I said, find the actual paper, use that to help you find the answer. Then explain what causes body temperature to rise during exercise. So the reason why whenever someone is exercising, the reason behind the increment in the body temperature is because of the process of respiration. So the respiration process becomes fast. so that energy is released in the muscles. Why do we need energy? So that there is enough ability for the work to be done. So this brings about overheating in the body, which makes the person to feel warm. So we'll say during exercise, so say during exercise, respiration releases energy in the muscles. Okay, then we'll say this brings about overheating in the body, which causes the person to feel warm. Okay, so now we move on to question C. So describe the effects of overcooling on the. So overcooling, we are looking at the body loses too much heat. So say the temperature receptors in this skin. So say the temperature receptors in the skin. So we'll say detect cooling and a signal. So there will be that communication, a signal in the form of impulse. In the form of an impulse is sent. To the hypothalamus. So this hypothalamus is like a structure that works like a thermometer. It's there to measure the temperature. Then it communicates with the other parts to bring about vasoconstriction and vasodilation. So say in the brain. Now when the hypothalamus is stimulated, then there will be that transmission of nerve impulses to the relevant body parts, which now brings about vasoconstriction. So we'll say this will cause the processes of vasoconstriction and vasodilation. So vasoconstriction is the process where the blood vessels reduces in size to reduce the flow of blood, while vasodilation is the opposite. So during vasoconstriction, the importance of this, we want the heat loss to be reduced, while for vasodilation, we want the heat loss to increase. Right, we have question 4. Figure 4 shows some of the plant and animal tissues. They are not drawn to scale. Okay. Now we have question A1. For each of the tissues A and D, state whether it is an animal cell or a plant cell. So if you look at this one, the cells are irregular in shape, so these are just the animal tissues. Then in cell D, we can see the regularity or the definite shape. So these are plant tissues or plant cells. Then you give reason for your answer in A1 above. So we'll say plant cell, so because of the definite shape and the presence of chloroplast. Then for animal cell, we'll say because of the indefinite shape and the absence of chloroplast. Okay, then what of question Roman number 3? Under Roman number 3, give the names of tissues B and C. So from the 4 tissues, 2 are for plants and the other 2 are for animals. So if you look at these structures, B, we'll say spongy. So spongy mesophyll tissue. Then for tissue C, we just say blood tissue. Then in which part of an animal or plant would you expect to find tissues A and D? So for tissue A, those are relay, they are found in the brain or spinal cord. Then tissue D, they are found in leaves, so just say the leaf. We have question 5. Both Mrs. Banda and Mrs. Walia delivered baby girls on the same day in the same hospital. Mrs. Banda's baby girl was named Anansi. Then Mrs. Walia's baby girl was named Wupe. Mrs. Walia began to suspect that her baby had been accidentally exchanged with Mrs. Banda's baby at the hospital. Blood tests were done on the 2 families. Right, so let's look at the results of the blood test. So for Mr. Banda, blood group is A. Then the genotype can be, okay, let's not consider letter I. So for A, we may have homozygous for A or heterozygous for A, or we don't know. Then for B, it's the same, homozygous BB, heterozygous BO. Then for someone with blood group O, the genotype is always OO. This one is just like this. Then Mr. Walia, since it's A, it can be AA or AO. Then Mrs. Walia can also be the same, AA or AO. Now, from this, we can see that there is a chance of these parents, of this mixing of babies. It's possible because AA cannot produce B, because from the alleles we don't have the allele for B. Now let's have a look at the questions that were asked. So we have been given these multiple alleles of the gene which control human blood groups. Use these symbols to answer the questions. Using full genetic diagram, show whether there was a mix-up or not. So genetic diagram is for Mrs. Walia and Mr. Walia. So let's begin. So begin with the parental phenotype. So we have Mr. Walia, then Mrs. Wadia. Now for Mr. Wadia, let's go to our table. Mr. Wadia, there are two chances. So it's AA or AO. We don't know, so we are going to make 2. Then from there we are going to make our conclusions. So here, let's say we use AA, then also here we use AA. So if we use AA, we move on to, so this should be phenotype. So we just say blood group A, blood group A as well. So move on to parental genotype. So let's say we use homozygous AA, we use AA, or maybe let's use the mixture. So we use AO, we also use AO, so that maybe we see the chance of having a child with blood group O. So we have a random fertilization. Before we put the random, let's look at parental gametes. Right, so we just have to separate these, then they become the gametes. From there we have random fertilization. So we can now cross. So we have AA, then this one will be AO. Remember, O is always recessive to O, so we begin with AO. So from this, there is a chance of having a child with blood group O. So say genotype of F1. Okay, now let's go to genetic diagram 2, Mrs. Banda and Mr. Banda. So begin with the parental phenotype. So for Mrs. Banda, from the table, Mrs. Banda is A. So we'll say blood group A. Then for Mr. Banda is blood group B. Then we'll move on to parental genotype. So even this one will say AO, then this one will say BO. Then from there we have parental gametes. We have A, O, B, O. Then random fertilization. So we can now cross. So there is a chance of having a B. There is a chance of having A. There is a chance of having B. There is a chance of having O. So say genotype of P1. Now for this couple, they have a chance of having blood group B, we can see. But for the first one, there is no B. So our conclusion is that this child, since he has B, belongs to the first couple, then this one belongs to this couple. That's the conclusion that we can make from here. So conclusion from the genetic diagram. So say, therefore, there was mix-up. Then from your knowledge of human groups, which blood groups shows codominancy? Is blood group AB. So AB is codominancy because the A is dominant and B is dominant, and from the two genetic diagrams, the one between Mrs. Banda and Mr. Banda is the one where we can have this condition. So that's it for this video. Thanks for watching.